So we're particularly interested in using genetic techniques in the mouse for dissecting neural circuits and the neurotransmitters that are involved in those circuits to control interesting behaviors or physiological processes. So anorexia, as you all know, is the loss of appetite, and it occurs in response to satiety. You've just had a big meal. It occurs in response to unpleasant stimuli, for example, an awful smell. You're not probably going to want to eat anything. Nausea is a good example where if you're nauseated, if somebody offered you a piece of chocolate cake, you'd probably say no thank you. If you're dehydrated, your interest in eating is very much reduced. In some kinds of cancer, appetite is reduced. And in anorexia nervosa and bulimia, young girls typically are more concerned with their body image and try to maintain very low body weight. So they have cognitive control over their eating behavior. Over the last really 15 years, there's been a tremendous interest in neurons that live in the arcuate region of the hypothalamus. And there is a population of neurons that make pro-opiomelanocortin, POMC. When those neurons are activated, they make a neuropeptide called melanocortin that acts on melanocortin-4 receptors in downstream nuclei. And that activation of that pathway inhibits feeding, and it stimulates metabolism. And that pathway is activated by a large number of inputs, especially hormones, for example, like leptin and insulin. And it's counteracted by the neurons that I want to tell you about, called AgRP neurons. AgRP neurons express this neuropeptide, Agouti-related protein, only in the arcuate, and we're going to take advantage of that. They also express neuropeptide Y, which was the original marker for these neurons, and they express GABA, the major inhibitory transmitter in the brain, which you'll see from my story is the critical neurotransmitter that these neurons make. So here you see that AgRP neurons are made reside in the arcuate, and they live right next to the POMC neurons. And they send projections to many different brain regions, from the arcuate to the parabrachial nucleus, which I'm going to talk about, but also to the paraventricular nucleus, to the nucleus tractus solitarius, many other outputs. So what I want to do today then is tell you about Agouti-related protein expressing neurons, and I want to concentrate on their projection, fairly minor projection, but a very important one, to the parabrachial nucleus. And I want to just basically use or describe our functional mapping technology, which involves the use of drugs and two viruses, adeno-associated virus and canine adenovirus, to map the circuitry. I'm going to tell you about the importance of GABA and glutamate in this circuit, and I'm going to tell you about an interesting adaptation phenomenon that occurs. Oops. So just as an overview, here's where we're going. The neurons, the AgRP neurons that live in the arcuate, send a projection to the parabrachial nucleus that lives right next to the locus coeruleus underneath the cerebellum. And what I'm going to tell you is that if you kill the AgRP neurons, that the parabrachial neurons become hyperactivated, and that somehow inhibits feeding, leads to severe anorexia and complete starvation. The idea is that the loss of inhibitory tone to the parabrachial leaves unopposed glutamatergic excitatory input to the parabrachial, and that's what's driving the anorexia. And the object of the last few years has been to try to identify the source of that excitation. So the goals of the talk then are to tell you that AgRP neurons are important, that they signal via GABA, they lead to hyperactivity in the parabrachial nucleus, and We're going to map the source of the parabrachial neuroactivity, and I'm going to tell you that the output of the parabrachial neurons is in fact glutamate, and the critical question is, where does it go? So AgRP neurons are important. A recent experiment by Scott Sternson at Genelia Farm used channel rhodopsin to selectively activate AgRP neurons. And as you can see on the right, when they turn on the light, activate the neurons, the animals increase food intake manyfold. You turn off the light, they stop eating. Brad Lowell's group at Harvard did the converse experiment. They expressed a modified G protein coupled receptor that they can activate with a synthetic ligand, CNO, and that inhibits these neurons because it's G-alpha I coupled receptor. And you can see in the red line that that inhibits food intake. Likewise, we did an experiment. We went into the arcuate nucleus and removed GABA selectively from the AgRP expressing neurons with a virus. This would be adeno-associated virus. And as you can see in the pink line, food intake decreased. When Serge Luque was in the lab, we targeted the human diphtheria toxin receptor to AgRP neurons, taking advantage of the fact that there are no other AgRP expressing neurons in the body. And so you can give diphtheria toxin, this big protein. You can give it peripherally, gets into the brain, and it kills all the AgRP neurons. The experiment you see here is taken, the figure you see here was taken a few days, probably about 6 days, after injecting diphtheria toxin, and you can see the complete loss of the in situ hybridization signal for AgRP. And coincident with that loss of AgRP neurons, if you see on the right, food intake gradually decreases and eventually goes to nothing, and not surprisingly, body weight falls. And if you don't intervene, the animal will starve to death. Interestingly, as they stop feeding, they stop initiating feeding, but even if you put food in their mouth, they won't swallow it. So they don't want to. So we thought it's as if they're nauseated, because if you were nauseated, you wouldn't want to go to the food, and even if you put food in your mouth, you'd probably spit it out. So here's the hypothesis again. We knew that this phenomenon would occur in mice that lacked AgRP and NPY, the two neuropeptides, so we speculated that GABA might be important. And the idea was that if we kill the AgRP neurons, there's the AgRP signal going away, that now you have unopposed excitation, and that would lead to the induction of FOS. So at this point, we collaborated with the Allen Institute and did whole brain in situ hybridization. I'm only going to show you one figure showing one of the outputs of the AgRP neurons, the paraventricular nucleus. And as you can see on the right, you get very robust induction of FOS when you kill AgRP neurons. So you remove GABA, and there's unopposed excitation, and you get FOS-induced. In fact, you could have mapped all the projection sites of AgRP neurons using this technology. So guessing then that GABA might be involved, Chi Wu, who just came to the lab a few years ago, had the idea of, well, let's try to promote GABA synthesis, GABA excitation of the parabrachial neurons by providing a benzodiazepine. Benzodiazepines are partial, this one anyway that we use, bertazinil, is a partial agonist, and it will potentiate any residual GABA signaling. And so he puts a mini pump into the mouse, then kills AgRP neurons, and what you can see is that in the black curve, the body weight begins to go down and then recovers over a period of 10 days or so, and likewise food intake goes in parallel. So they actually recover. And interestingly, at the end of the experiment, the mini pump is run out. At that point, the mouse will continue to eat just fine. So it has adapted to the loss of AgRP neurons, but it takes about 10 days. So we've basically provided some protection of these critical neurons. We don't know where they are in the body yet at this stage, but providing an extra GABAergic inhibitory input someplace in the brain is providing protection against severe anorexia. So the question is, where does that occur? So she modified the protocol, and instead of delivering the bertazinel systemically, he has a pump on the back of the mouse, and he has leads that go in to 5 different brain regions and ask where bilaterally will bertazinel protect against anorexia. And as you can see, the curve labeled PBN worked. You get this dip and then a recovery, and the other 4 brain regions all led to starvation. So that was our first indication that there's something about the parabrachial nucleus where GABA signaling can prevent the severe anorexia. Whoops. We also did an experiment where instead of injecting diphtheria toxins selectively or systemically, we inject it selectively in the parabrachial nucleus, and that also leads to severe anorexia. So the strategy then, or the idea is that AgRP is providing an inhibitory input into the parabrachial nucleus. You remove that and you get anorexia. So then we wanted to map where is the excitation, where's X in this figure. And so the idea was that if we could inhibit either the input to the parabrachial or the output of the parabrachial, we should be able to prevent the anorexia. And you're going to see in the next few slides that that strategy works. To give you a preview of what I'm going to tell you, we think that there's a glutamatergic input that's providing the direct excitation. It's coming from the nucleus tractus solitarius, and it's acting on NMDA receptors in the parabrachial nucleus. The NTS, in turn, is activated by serotonin, acting on 5-HT3 receptors, an ionotropic receptor in the NTS. And the output of the parabrachial nucleus is glutamate. So the next few slides, I'll show you the evidence that substantiates that little circuit. So remember I mentioned that when these mice stop feeding, even if you put food in their mouth, they won't swallow it. And that gave us the idea that this is sort of like a nausea pathway. And there's good evidence in the literature that nausea signals from the vagus do in fact impact on or are transmitted from the NTS through to the parabrachial nucleus. So there was some anatomical reason to suggest this as well. The point is that if we introduce an antagonist, in this case ondansetron, of a serotonin receptor, and we do it in the nucleus tractus solitarius, but not in the PBN, we can get the same kind of dip in feeding, body weight, and then a recovery. So the next thing we wanted to know was where is the serotonin then coming from that's acting on the serotonin 5-HT3 receptor? For this experiment, we begin to use viruses. We're going to use canine adenovirus, which has the nice property that it's retrogradely transported. So we have a mouse that has TPH2 conditional. It's functional, but it can be inactivated by the action of Cre recombinase. So you can inject CAV Cre into the target. We know the NTS is the target. And then we can let it inactivate TPH2, all the serotonin coming from the neurons that innervate the NTS should now be removed. And then we can kill the AgRP neurons with diphtheria toxin and see what happens. And as you can see, they recover. And what's nice then is you can go back and ask, okay, where have you lost TPH2? Where you still have other markers of serotonergic neurons. And the answer is in the ventral raphe nuclei that are in general known to innervate hindbrain structures. The next experiment, we're going to explore the connection between the nucleus tractus solitarius and the parabrachial nucleus. We're going to make a guess that it's glutamate, and we have a mouse that has a conditional VGlut2 allele. VGlut2 is the vesicular glutamate transporter that's necessary for getting glutamate into vesicles so that it can be used as a neurotransmitter. We're going to inactivate VGlut2 in the nucleus tractus solitarius and ask whether you rescue the animals from severe anorexia when you kill AgRP neurons, and the answer is yes. You can also do the same experiment by going into the parabrachial nucleus and knocking down really one of the critical glutamate receptors, the NMDA receptor, and that also protects. Oops. Boy, this is really... If one goes into the parabrachial nucleus itself and takes out the ability to put glutamate into vesicles using the same VGlut2 conditional mouse, one can also protect against anorexia, suggesting that the output of the parabrachial nucleus that's critical is in fact glutamate. And in this experiment, it's kind of shows the converse. If you take out VGlut2 from the parabrachial nucleus, but you don't kill the AgRP neurons, now you actually get weight gain. Okay, so we're beginning to get the idea that too much activity of the parabrachial nucleus inhibits feeding and leads to anorexia. Too little activity actually promotes feeding. So here's kind of the final model. We have the parabrachial nucleus, which can be activated by a number of inputs. I've only concentrated so far on the NTS input, but there are probably cortical inputs that directly or indirectly impact here. There are gustatory, all taste stimuli, vagal stimuli, all come through parts of this pathway. Dehydration probably impinges on the parabrachial nucleus. And so, just as I said a minute ago, our model is that too much activity of the parabrachial inhibits feeding, too little stimulates feeding. And so here's the pathway once again. Basically, there's an inhibitory input from the arcuate that's probably sensing overall metabolic balance, how much energy reserves the animal has. Then there are inputs from the periphery and sensory systems that are coming in through the NTS, and they're meeting in the parabrachial nucleus to influence feeding behavior. And the pathway that I've just shown you is shown at the bottom, where we've used genetic techniques and viruses that are either retrograded, transported, or acting directly to manipulate the production of important neurotransmitters to affect an interesting behavior. And so there are other inputs that need to be mapped. We're currently in the process of doing that. And for the future, the parabrachial nucleus, like the cortex that you heard about today and other brain regions, is incredibly complex. There are lots of different neurons in there, and likewise in the NTS, lots of different neurons. We need to identify the specific cells within the parabrachial that are mediating the anorexia. Currently, the only marker we have is they become FOS positive when you kill AgRP neurons. What markers do they actually make? And once we have those molecular markers, then we can have Cree driver lines and then get much more sophisticated about what's actually going on. A key question is, what is the output of the parabrachial? Where does it go? We know it's glutamate. Where does it go? And how does it actually lead to anorexia? What are the direct and indirect targets? And I've kind of glossed over, there's this interesting adaptation phenomenon. If you kill AgRP neurons and you can protect them with either bertazinil, ondansetron, or these viral approaches, about 10 days later they adapt. What's going on there? What kind of adaptation are we talking about? Is this rewiring or changing in synaptic plasticity, changing in glutamate input, GABAergic input to get it back in balance? How does it work? Until we have molecular markers and can look in specific cell types, it's going to be very difficult to answer that question. But it's a very key one, I think. And so, finally, this work is started by Serge Luquet, and Chi Wu has done much of the work that I talked about today. We've had some help from several people in the lab, in particular Maureen Boyle here at the Allen Institute helped us identify the parabrachial nucleus as being really critically involved. Supports come from HHMI, the Klarman Institute, and NIH. Thanks.